Go ahead.
Okay. Now, good morning, everyone. My name is Jason Ofosu-Mensah, a student at Bronx Community College. And today, my presentation is titled A Bioinformatics Approach to Understanding the Clinical Relevance of the Proprotein convertase with furin in Cervical Cancer. Well, what is cervical cancer, firstly? Cervical cancer is a type of cancer which occurs in the cervix, which is the lower part of the uterus, which connects to the vagina. HPVs and STIs also play a role in causing most cervical cancers. So the symptoms of cervical cancers usually don't start are not known in the earlier stages, but as the cervical cancer advances, we could see that there's bleeding after intercourse, douching, menopause, or pelvic examination, and so on. And there are four stages of cervical cancer. And in the first stage, there are four substages. And we can see that in the first substage, the cervical cancer cell starts out as a small pencil line, but in the other substages, it starts to metastasize or grow throughout the cervix. And in the second substage, there are two substages. And we could see that in the substages, we could see that it metastasizes from the vagina all the way throughout the uterus. And in the third stage, there are also three substages. We can see that the cervical cancer metastasizes from the cervix through the vagina, all the way throughout the kidneys, and also into the lymph nodes. Also, in the final and the fourth stage, there are two substages in which we can see that the cervical cancer cells metastasize from the cervix all the way throughout the liver, the lungs, and the lymph nodes. Now, I'd like to give it up to my co-presenter.
So my name is Zouberou Sayibou. I'm also a student at Bronx Community College, majoring in computer science. So we are studying the relevance of furin, which is a main purpose of this project. So furin is a protein convertase, which is also known as an enzyme that activates other genes in our body. And we are going to see how it activates cervical cancer using bioinformatics. So we collected, what we have done so far is that we have collected some data sets from the Human Protein Atlas, and then also we downloaded this and uh, conducted some descriptive analysis. And we also use the shrink pathway analysis, which is a website that generates predictive model that shows the protein genes that may be directly or indirectly affected with the furin. So when we collected the data set, we have 291 patient data set, and then 230 of them are alive, and 71 of them were dead when the data was collected. Based on the ethnicity of the data collected, the 70% of them are white, and then 10% were Black or African American, and the rest are being shown. So as I said, furin has a direct, indirect pathway analysis with other genes. And this diagram shows that, and these are some of the genes that that furin can can activate in our body. So the survivor analysis, which is the Kaplan-Meier survivor analysis, which is a a statistical model used to study data sets. One of the uh, really used to study data set in biology. So as our data set is showing, as our diagram is showing, we can see that the lighter color shows a higher furin expression, and the lower color, the darker color actually shows lower furin expressions. And then we can see the number of years from zero to we have from 0 to 15 years that it shows, and it has shortened the lower furin expression, which is the darker color, which shows a higher ages. So what does that mean? It means that the higher 29.75 expression of 29.75 is the number of furin expression, and that is a higher number. And then less than that is a lower number. And from our data set that we collected from the Human Atlas, 40% of them have higher, and then 60% have lower. So what can we say from what we have just discovered? So based on our data set, the majority have less fluid expression. That means most of the patient that the data set were collected have less furin expression, and the patient that have less furin expression tends to live longer, and patient that have a higher furin expression tend to have a shorter lifespan. So this means that if we can find any ways that we can shorten the furin expression in our body or in the human body, then patients that have a cervical cancer will have a higher um, they'll have a higher lifespan than people that have a higher furin expression. So the direction of this project is to actually search for drugs that will inhibit furin expression, test those drugs on a group of cervical cancer cell lines. So this is our direction of the project, and this is what we are looking forward to do. If we can find these drugs, then there is a possibility that there is a cure for uh, the cervical cancer. Thank you so much.